Okay, Dr. Koshy, it is all yours.
Okay, so my name is Anita Koshy, and I work in neurology and in immunobiology, which is our micro and immunology department. And I work on a parasite called Toxoplasma gondii, and I like to think that we think about how toxoplasma affects your brain, and I'll explain why that matters in a moment. Okay, so this is the parasite that I work on. Can you see my cursor? Yeah, great. Okay, so it's a eukaryotic, so single cell organism that has a nucleus and is related to the parasite that causes plasmodium. Related to the parasite plasmodium that causes malaria, which you've probably heard about. It's a very different parasite, but they're related. And the thing to know about Toxoplasma from this picture, very small, about 2 microns by 10 microns, but it has these unusual secretory organelles, so things it can send out proteins with. And these are the rope trees, which are right here, and the dense granules, which are these dark areas here. And Toxoplasma, like most, sorry, let me turn off my email because it's going to drive me nuts. So Toxoplasma, like most obligate intracellular parasite microbes, which is what this is, needs to depend on that host cell. And so those secretory organelles will become important in a minute. But the reason we think about Toxoplasma is that it infects a lot of people. So Toxoplasma infects about 1/8 to 1/3 of the world. And what happens for most people is they eat their Toxoplasma, or they ingest their Toxoplasma either through contaminated food or water. And it starts in one form, either called a sporozoite or a bradyzoite. And in the gut, it then flips into this other form called a tachyzoite. The tachyzoite form then goes and disseminates across the body. The immune system responds to it and generally takes care of the tachyzoites. But a small population of those parasites will flip into this slow-growing form called the bradyzoite. And this slow-growing form will persist in specific cells in the body and persist for, we think, a long time, so up to the lifetime of the host. And so the thing that's important about this persistence, which can happen from anything from birds to humans, is that it enables the parasite to cause symptoms later or cause problems. And so the places that Toxoplasma insists are the heart, skeletal muscle, and the skeletal muscle, and then the brain. And the brain is where we're interested. And for both humans and rodents, the brain is a major organ of persistence. And that is great for us because it means that we can use mice, and they're actually a natural model. And that has some really large advantages. So why do we think about the brain? Because the brain, or the central nervous system, is the key to symptomatic disease. So this is an MRI that I pulled from the internet. It's a normal person's MRI. If you have a normal immune system and you happen to have Toxoplasma, this is what your MRI looks like. I can't tell any difference between you and someone with or without Toxoplasma. If you're unfortunate to be a fetus when a mom gets Toxoplasma, especially in the first trimester, then you can have some really terrible consequences in your brain, which should look a lot like this. This happens to be a CT, so it looks a little bit different, but it should look very similar to this, is obviously not. And so that's, Toxoplasma is the reason women are told not to change kitty litter, is because the cat is the definitive host and will poop out a huge amount of Toxoplasma when it first gets infected. Okay, so let's say you don't get your Toxoplasma when you're an infant, you get it and you're here, and then you are unfortunate, you develop either AIDS or you get an organ transplant. So then your Toxoplasma can reactivate. And again, in the brain, this is an AIDS patient I saw when I was an infectious disease fellow. And all these areas that you see some white around, that white is dye that we've given the patient. These are all basically abscesses. And it's basically the Toxoplasma has reactivated, and now is the immune system is trying to keep it from causing too many problems. And so it's sort of walling it off. And so our big question, so the CNS is a key issue for people with Toxoplasma. And so our big question is, how does Toxoplasma persist in the CNS? Why, how, where? And so our big hypothesis is that it depends on its ability to manipulate the host cell. And again, we think that because Toxoplasma has all these organelles where it can shoot in proteins into this host cell to manipulate it. And so the way we've studied this is we created a system called what I call the Toxoplasma cruzi system, where we can identify what cells are interacting with Toxoplasma. So our parasite shoots in a pair. We've taken one of those proteins, we've connected it, so one of these rotary proteins, and we've put another protein called Cre recombinase on the back end of it. And in specific mice, that Cre recombinase can go into a host cell and cause the host cell to now express green fluorescent protein. And so it looks like this. Our parasites are usually red. The parasite comes into contact with, I'm going to show you a neuron. It's going to inject in the Cre recombinase. That cell is going to turn on green. And so now, because it's a genetic change, we can follow that change in that cell for the rest of its life, because we'll always be able to see this green protein. And the way that looks, we call these Toxoplasma injected neurons because you're getting injected with this parasite protein. And this is the way it looks. So we can see all these green cells, and we can see that they're neurons because you can see that they have these long processes. Only one cell type in anyone's body that has long processes like that. Those are neurons. And so we're really interested in understanding why Toxoplasma interacts with neurons, how it interacts with neurons, and what the effect of that interaction is, both on the parasite and on the brain. And so that's what we do. So what is this parasite doing in your brain? So we really try and understand these ones that have been manipulated. We've done RNA-seq on them using a system called laser capture microdissection. We'd like to do now single cell RNA-seq on them. We've shown that these neurons can respond to interferon gamma and can kill parasites. And actually mouse neurons, at least in a dish, are less efficient at blocking Toxoplasma than human neurons. So we've done this both in mouse and human neurons. And in the setting of interferon gamma, human neurons seem to be able to resist Toxoplasma much better. And then we've done electrophysiology. I'm obviously not going to go into all the details here. These are really hard things to do, but what's interesting is, so this green neuron that's been injected with Toxoplasma protein and then turned green, if you do what's called electrophysiology, where you look at the neuron and the different sort of electrical signals it'll send and where it stands, the neurons that have been injected with Toxoplasma protein are super abnormal. And their resting membrane potential is way higher than it should be, so they won't fire normally. But if you look at neurons that are about 100, someplace between 30 to 100 microns away, so 3 to 10 cell bodies away, maybe up to 12 cell bodies away, they actually look very normal compared to the same neurons in an uninfected mouse. So there's something very specific about this interaction with Toxoplasma potentially, or whatever's causing these neurons to be abnormal is extremely tightly regulated. And either of those are pretty interesting to us because they have implications for immune responses in the brain in general. And so we also manipulate the parasite and are looking into why toxin primarily persists in neurons. And can we understand the neuronal genes and the parasite genes that are allowing it to do that? Okay, and then some quick skills from the Koshy lab. You'll get, if you end up in our lab, you'll get a lot of immunohistochemistry, immunofluorescence, so sectioning brain, staining it, and then quantifying that staining using light and epifluorescent microscopy. We do a lot of molecular cloning, so engineering parasites, so knockout lines, ectopic expression, and then validating the parasite line. And then science, what I like to call soft skills. So you learn to give scientific presentations. I think of it, lab meeting is a very gentle place to sort of learn, but we'll all critique you. And then learning how to read scientific articles. So we'll do a lot of journal club during summer. And with that, I will stop. So these are the people in my lab. This is us. A summer day. Hannah has gone on and graduated. These are my two most recent UBRP students. Chloe is off to Scotland this summer. And so we are definitely looking to add someone for the summer. And with that, if there are any questions, I'll be happy to answer them.
All right, students, if you have any questions, then feel free to unmute and ask them.
I'll tell you that.
I had a,
Yeah, go ahead.
So I had a question on Is there any natural way that the neurons respond to these parasites, like a way to get rid of them?
Yeah, so
Or-
all of it is interferon gamma dependent. So in general, most of your cells in the body need interferon. So not say maybe macrophages, but pretty much every other cell in your body that's not sort of a professional killer cell needs interferon gamma, which is a cytokine, to tell that cell to upregulate a bunch of stuff. And once it upregulates a bunch of stuff, that stuff that it's upregulating will kill the parasite. And it does that not with 100% efficiency, and it does it in certain cells better than other cells. And we think that's how you end up getting persistent parasites. Does that make sense?
Yeah, thank you.
All right, students, any other questions for Dr. Koshy?
If not, I will stop sharing. I hope you guys have a good rest of your day.
All right, perfect. Thanks for joining us. I appreciate it. All right, so faculty number 2 on the list is Dr. Giovanni Melandri, who is here from Plant Sciences. And I think I gave you sharing powers. Are you able to share your screen?
Okay. Screen 2. Can you see the screen?
Yes. All right. Yeah, the floor is yours then.
Okay, thank you. So yeah, well, thank you for giving me the possibility to show you a little bit of what I'm planning to do into my lab, considering that I'm a new faculty and I started in January this year. So yeah, my name is Giovanni Melandri. I'm an assistant professor at the School of Plant Sciences. And in my lab, we will leverage physiological and biochemical data to improve drought tolerance in crops. So the research program is a kind of systems biology for crop improvement in arid environments. So we will have experiments conducted at the University of Arizona Maricopa Agricultural Center, where we will grow crops in there. We will collect physiological data on these crops and the performance data. As you can see here, there is like the gantry system in there, but we will use also handheld instrument to measure like photosynthesis, fluorescence, so parameters, lichens, stuff like that. On these experiments where the different germplasms, so different genotypes of different crops will be evaluated, we will also sample leaves. And these leads will be used to collect omics data on them. You will see also in the next slides, I'm particularly interested in metabolomics data, but we can also collect transcriptomics data. And if we use different genotypes, especially diversity panels or mapping population, we can also use like genomics data, so SNPs, to conduct some other analysis. So the analysis that we're going to do on this data are mainly machine learning models for trait prediction. So we will try to use the metabolomics variables and the physiological data to be variables used in models to predict trait performance. And these models can also give us like the best biomarkers, so the best variables that are like our targets to improve crops. Because if it's, for example, a metabolite, we can then investigate the biochemical pathway that is like behind that particular metabolite. But as I said, if we have larger panels, diversity panels or biparental mapping population, we can go for GWAS and TTL mapping. So we can even use this as quantitative traits to do association mapping. And these objectives from 1 to 4 are going to be covered by my lab already this summer. I will have like field experiments and the lab to perform this analysis. But in the long term, we will use these targets to edit candidate genes and finally validate in the field if we can really improve the performance of these crops. We are in Arizona. It's a desert. It's a semi-arid environment. So this means that basically we're not going to have drought or strong stress just in specific phenological stages. We're going to have drought, very hard stress basically throughout the crop cycle. So we're going to be in this situation. And this means that we have to work with crops that can actually are actually able to survive these conditions. So my program will cover crops that can be grown in the Arizona low desert. crops that are of interest for the US and Arizona agriculture, and crops for which we have like genetic diversity available. Otherwise, we cannot really find the differences between genotypes and the secret of the difference in being able to be drought tolerant or heat stress tolerant. So I will work on cotton. That this is a picture I took in the field some years ago. And we will work with sorghum too, which is a very drought tolerant cereal crop, very important. Also, you will see it will expand a lot the cultivation of sorghum because it's one of these cereal crops for the future. And in the little bit longer term, I will also try to work with durum wheat because among like the wheat is also soft wheat. Durum is much more tolerant to abiotic stresses. And I said we're going to collect omics data on the samples, on the leaf sample of these crops. Why I'm interested in omics? Because we have to find a way to get a cellular phenotype, so an endophenotype that can link the genotype to the phenotype. And you know that there are different layers of regulation from the genome to the phenotype. So the endophenotypes layers of regulation are like molecules like DNA, mRNA, proteins, and metabolites. But you can see that basically the metabolite is the closest one to the phenotype. So it's a kind of integrator of what happens at the DNA level, at the RNA level, the protein level, and finally you get a measure, a metabolic value that is a kind of integration of all these regulation levels. Plus, the metabolite is also having a strong interaction with the environment, more than the other layers of regulation, but let's say not more, but it's the fact that it's at the bottom of this steps, right? It's like integrating all the environmental interactions. So it's the one of the endophenotypes that is closer to the phenotype and can direct us backwards until the DNA level. And so what's the connection that we have between drought stress and plant metabolism? Well, everything starts at the leaf level by the closure of stomata that are inducing a difference in photosynthesis that is then inducing in turn a change in metabolism. So basically, metabolism is changing under drought to maintain essential function and adapt to stressful conditions. Plus, a new set of molecules are produced to face the stress. And in my lab, what we're going to do is to measure the metabolic changes with mass spectrometry, especially in this case will be untargeted mass spectrometry. So we're going to try to cover as many molecules as possible. But you can say, you see there in the leaf picture that there is something called the ROS scavenging. So ROS are reactive oxygen species. So another thing that happens when you close the stomata is to have an excess of absorbed light energy that cannot be used for photosynthesis anymore. So you produce pro-oxidative stress conditions. And there is a strong antioxidant systems in plants that counteract, try to scavenge this ROS. And we're going to use spectrophotometric assay to measure enzyme activities of this antioxidant system and also non-enzymatic antioxidants. So basically, if you join the lab this summer with the UBRP program, what you're going to do in my lab is help with some of the cotton and sorghum experiments in the field when we're growing the plants. At the same time, you will measure metabolism and oxidative stress in the lab. And then when you collect all this data, we will have like to normalize, impute, correct the data, and also model this data for the prediction of some of the traits, or at least see if they can direct us into some of the metabolic pathways that are more interesting for drought tolerance in the crops that I have just described. And yeah, well, you have any questions? I'll be open to answer to you.
I wanted to ask a random question, since you have so many different types of techniques and the different settings for your research is quite diverse in terms of being in the field and being in the lab and analyzing data. What would an average day look like for an undergraduate in your lab?
That's a good question. I have hired a postdoc. This guy is the guy that is taking care of the lab. So we can also discuss with the undergraduate students if they prefer to have like more like physiological and field work. So I can direct them more to that, or if they want to do more work in the lab, so that's fine. They can work more in the lab under supervision also of this postdoc that I just hired, or if they are more interested into the let's call it bioinformatics part, like so coding a little bit using R to run the models for machine learning. That also can be an option. I would say that probably the field part is the hardest one, also because it's in Maricopa. So I guess that probably the majority of the undergraduate students are going to be interested into these components here, the bottom two more than the top one. But of course, they are more than welcome to come to the farm, at least to help, I don't know, once per week or see what happens in there. That's totally fine with me.
Thanks. Students, what other questions do you have? Okay, I feel like I have to wait at least my obligatory 15 seconds. I think we're good on that. So if any of you are interested in working with Dr. Melandri, then feel free to reach out to him. And thanks for being here today. I appreciate it.
Thank you.
All right, and up next, we have Dr. Carini, who I'm going to get set up here. Hello there.
Good. See
Hi, nice to see you. I think I gave you sharing powers, so hopefully you have the ability to share your screen.
Okay, let's see here.
All right, looking good.
Looking
All right,
good.
so the floor is yours. You got up to 15 minutes as far as presentations and Q&A.
Okay, awesome. Thank you so much. Thanks for the invitation. I'm really excited to share some of the research opportunities and some of the research that we do. I'm in the Department of Environmental Science, but I have affiliates in MCB and the genetics program and plant science and all sorts of other places. We're microbiologists that do systems biology type research to try and understand microbial ecology across different environments. So why should you care about microbial ecology? Well, microbes coat all of us. They're inside all of us. You're probably sitting at a desk. They're all over the desk. They're on your phone. They're pretty much everywhere, including in the soil, in the water, in the air, as we all know from the last couple of years of the pandemic. So trying to understand how they interact and what controls their distributions is something that's really important to understand how the Earth functions and how we can make Earth habitable to humans and other forms of life. So we don't actually have an ecosystem that we study. We just study microbial systems biology across different ecosystems, and I'll go through that here. If anything that you're interested in, if you hear anything you're interested in, I'll put this QR code up again at the end of the talk. Just scan it. That'll bring you to our website. And there are some QR codes throughout the talk that might interest you, so have your phone handy to scan those. Our research lab right now is pretty small. We're going to grow this fall, and hopefully you'll be a face that's on here as well. We've got two researchers in the lab, Dr. Snow in Boast West and Christina Bond. They're carrying out the bulk of our funded projects currently, and we have Michaela and Coralie, who are two undergraduates in our group currently that are assisting Christina and Una to do their research projects. And then we've had a whole bunch of different students with us in the past. They're not all listed here in the lower left. Izzy Viney was a recent master's student. We've had some rotation students, and last summer we had a UBRP student. Actually, that's a typo, not an RU student, Brooke, who did a fantastic job looking at desiccation tolerance in some soil microbes for us. We're funded by a variety of folks listed here. We've got NIH funding to investigate arsenic tolerance in mice. NSF funds, as well as the Simons Foundation funds our work. So just to explain a little bit about what we do. So these microbes are everywhere in the environment. You're ubiquitous in all places on Earth, from the atmosphere to the deep ocean. There are two schools of thought on how to study them. One is what we would call top-down approaches. The way I like to describe this is it's the equivalent of taking a snapshot from a drone of a forest, where you can count all the trees and count all of the animals in that photograph and get an idea of the community ecology of that environment. Well, we can do that at the molecular scale now using a variety of techniques that are listed here. Microbiomes you may have heard of. That's just one of several different approaches that gives us this high-level community type analysis. The other side of this equation is what we would call bottom-up approaches. And this is more traditional microbiology. And when you think about microbiology as a discipline, you might think of things like culturing microbes in the lab and studying them and how they react to different perturbations in the lab. We call this systems biology. And what our lab tries to do is bridge these two extremes by using systems biology, bottom-up type approaches to really get at an understanding of the actual microbial ecology. The top-down approaches on the left here are very coarse. They've exploded the field of microbiology in the last decade, but they are still very coarse. It's really hard to identify specific microbes that are performing specific features in any given environment, whether that's our gut or a soil sample. On the other side, systems biology approaches are very good at that. You can culture a microbe, you can construct an experiment, test a hypothesis, and know exactly which microbe is doing exactly which function. The problem is we can't grow most of these microbes. So our lab puts a lot of emphasis on trying to figure out how to grow a lot of these microbes. So it's a very reductionist, small scale vision. We kind of work somewhere in the middle in what I like to think of as microbial systems ecology, merging systems biology and microbial ecology together. So how do we do this? Well, shortly after I started my group here 6 years ago, we developed a culture collection of what is now over 3,000 different microbial cultures from local environments. So our sampling sites are kind of listed here on a cartoon. They're from the Santa Catalina Mountains just outside of Tucson. And this photograph displays that nice. We have Tucson here down at the bottom, and then the Santa Catalina Mountains all the way up here. We have different sites that span that sort of mountainous gradient. So there's differences in temperature, differences in precipitation, differences in soil structure and the way it moves through the ecosystem. And we just did a lot of microbial culturing our first 2 years. And we have all of these cultures now that are from very different groups of microbial taxa, but all of them are pretty important to the ecosystems that are local to us here in Arizona. We've selected a few of those and developed them into model systems to try and understand their adaptations to some of the local stresses. All of you probably know one of the big environmental stresses here in Tucson is desiccation and heat, right? It gets really hot, really dry in the summer. And we've spent a lot of time trying to understand how microbes that don't form spores, so they don't form a protective coat to help cope with that, how they survive in soil when it's when the soil temperature is 120 and there's essentially no water available. This is one of the types of projects that a UBRP student would participate in, one of the types of experimental analyses, where we test desiccation tolerance by using flow cytometry, which counts individual cells, as well as traditional auger plate plating techniques to try and ascertain what the culturability or what the relative survival of these strains are to desiccation. So we filter them down and then dry them out and then try and assess how many cells come back to life after we've dried them. We did this with a UBRP student last summer, and found some really interesting data that even very closely related microbes, so these are things that most microbial ecologists would call the same thing, have a tremendous variability in how tolerant they are to desiccation. Here we have different strains of bacteria. They're all Bradyrhizobium, which is a very common soil microbe. And then we're looking at how likely they are to come back after drying out. If it's 100% relative culturability, that means that everything survived desiccation. So none of them quite hit that point, but many of them are quite desiccation tolerant. And those happen to be from our desert sites, which is interesting. So this is a type of experiment that you would learn how to do in our lab, should you decide that you want to join us. We also have some other projects that are just sort of getting their feet. And this is something that I recommend you scan with your phone. It's a cool article that the Arizona Republic did about the research that we're doing, trying to culture archaea from the desert. So these are a type of microbe. They're very similar to bacteria that inhabit soil layers that are just below the surface. So about 20 centimeters below the surface of the deserts local to us. And they use ammonia as an energy source. So we eat food, we eat carbohydrates, we eat fats, and we use that as an energy source. These microbes don't do that. They eat ammonia, and we're unclear where that ammonia comes from, but we know they're abundant in the environment. And now we have pretty convincing evidence that this sampling trip that this article discusses resulted in some cultures of these for the first time from the Sonoran Desert. So we're really excited about that. Right now, Michaela is working on that. She's an undergrad in our lab. This could also be a project that you participate in as well. We're very excited about this, and surprised we got the culture, to be honest with you, but we definitely have it in culture now. Some of our other projects that are getting started right now, we work with deep sea microbes. So a collaborator of mine isolated some microbes from the deep ocean that were deposited in the seafloor, on the seafloor 6 million years ago. To give you a perspective of when that was, humans had not yet diverged from their ancestors evolutionarily when these microbes were deposited. His group revived them, resuscitated them from these deep sediments that he collected. We published a paper about it last year, 2 years ago, I guess now. And we have these cultures now, and we're trying to mimic in the laboratory these really slow growth rates that those microbes typically would grow at when they're in the seafloor. So the doubling time, or the time it takes for those cells to divide once, is 10 to 100 to 1,000 years, depending on where they are in the seafloor. And we're trying to mimic that very slow growth rate in the lab. So in essence, they're not growing in the lab. They're just sort of hanging out. We're doing that by building a special cultivation device. So if you're someone that likes working with your hands on maybe electronic devices, or is somewhat mechanically minded, but also has an interest in biology, this might be a project that would be really, really cool for you to participate in as well. So what can you expect to learn from us if you decide to do a rotation with us, or do spend a summer with us? Skill development is one of the big things that we try and help undergrads with. Things like the polymerase chain reaction, nucleic acid extractions, different assays. You'll probably grow bacteria and prepare media for bacteria, flow cytometry. We train our students on, our undergrads are all doing that, I think now, as well as some project-specific techniques. You can also expect that we'll mentor you intentionally. You're not just an undergrad in the lab. All of the people that mentor undergrads in our lab are trained in mentoring. So they've either taken a class or they've gone through some sort of program to be a quality mentor. We try to individualize our mentorship for you and what you need and what you're hoping to get out of the experience with us. And I always like to highlight this, that we care about you first and your work second. So we care about how you develop as a undergraduate and a potential researcher first, and the quality of work, that's something that improves with time, and we understand that. And it's okay if you make mistakes, and we in fact expect that from most of us, from most undergrad students. You can also expect to have some fun. So sometimes we take field trips. This here is from a trip to Mount Lemmon last summer to send Brooke off. She was a visiting UBRP student from Michigan, Detroit, Michigan. So we took a trip up to Mount Lemmon and just spent an afternoon up there playing games, and had a lunch. And it was a lot of fun. Sometimes we do field excursions. We are planning to go to Mount Lemmon in late May. I think that probably won't coincide with the start of the UBRP program, but we occasionally do things like that. Sometimes we'll go out to the desert to collect samples and things like that. Always happy to bring people with us on those types of trips. So that's all I have. I'm maybe a little bit early here, but I'm happy to entertain any questions you might have, and either right now or by email. Thank you.
Thanks. All right, students, any particular questions you would like to ask? Okay, good job then.
Thank you.
So yeah, be in touch with Dr. Carini if you're interested in his work, and thank you for being here today. I appreciate it.
Yes, no problem. Let me stop my share here.
All right. Thank you. And last but certainly not least, we have Dr. Jean-Marc Fellous, who is here from the psychology department to share his research as well. How you doing, Jean-Marc?
Hi, Jennifer. Good. How are you?
Good. Nice to see you. I think I have given you sharing powers.
All right. Let me try.
You can go whenever you'd like.
Let's see what happens. All right. Can you see my presentation?
Yes.
All right. And if I do this, can you see it still?
Yes.
Great. Thank you so much. I really appreciate the opportunity to come and talk to you about what we do and potentially recruit, of course, you guys, students. So I am the director of the Computational and Experimental Neuroscience Lab. And we have all kinds of projects. I just wanted to list them here so you know a little bit about what we do as a lab, but the one that I'm going to expand on and we need help with is this one on multi-goal complex spatial navigations in rats. All right, so I'm a rat person, I do rats. So briefly, this is what the theme is about, spatial intelligence. All right, how are you smart with space? How do you navigate efficiently in complex space? That's the key here, that we're really interested in complexity. We're not really interested in very simple things like turn right, turn left. We want to challenge the rats, right? How do you remember navigation efficiently? How do you improve your ability to navigate in space? And one thing is also that we do is looking at this during sleep. It so happens that sleep is very important for learning and for consolidating and for improving. All right, so essentially, can you see my pointer? Thank you. So essentially, this is what we're interested in, right? So complex spatial navigation with all kinds of obstacles and challenges. And we're hoping for the rats to do something like this. So essentially find creative solutions to complex problems, all right? And of course, go into the sleep part, which we think is very related to this ability to navigate in space and to find interesting solutions to spatial problems. All right, so very briefly, I'm sure you've heard of all these terms before, hopefully some of you have. There is, of course, a brain area that is special for spatial navigation that has been identified. So we know exactly which part of the brain is involved, or several parts of the brain, but the hippocampus is this structure that is specialized for spatial navigation. So as I'll show you in a minute, we record from the hippocampus while the rat is doing a task. Okay, so we're interested not only on behavior, but on the ability of the brain to code, to predict, to do this at the neural level. So what do we know about hippocampus? Well, they contain place cells. Place cells are these special neurons that tend to fire to become active in a particular location in an environment. So this is an example with a square box where the rat is running around. And then here is the square box, or multiple instances of the square box. Each of them is showing one cell. So this is a square box where there is no cell active. This is a square box where there is a cell active pretty much everywhere. And then you have, from time to time, these weird cells that are actually active only in a particular place. All right, those are the place cells. Those are the ones we think are really important for understanding space. Of course, you may know this, the Nobel Prize was given for that discovery in 2014. In a way, our work is a bit of a follow-up on all of this fantastic work that has been done. Now, here is the thing, is that all this work, 99% of all this work on place cells that led to the Nobel Prize were done on very, very simple environment, very, very simple task. All right, so the rat has just run around, that's it, no task, that's this. Or go straight, turn right, go straight, turn left. This is it. All right. So we want to go beyond that, essentially, in the lab. We want complexity, right? We want something complex. There are many ways of being complex, but believe it or not, we have the largest maze in the world. It's downstairs in the building. It's called the megaspace. It has these dimensions here, so pretty big. We're recording rats doing tasks in this, and I'll show you a movie in a minute. Using wireless techniques. So we hook up the rat to Wi-Fi and they become Wi-Fi neurons. So essentially we listen to the neurons while the rat is running around using our Wi-Fi. And then we, of course, complicate things. We make things interesting for the rats. Not only is it big, but we can do all kinds of things in there. We can put walls, we can put, as I'll show you in a minute, robots. We can do all kinds of things to challenge the spatial navigation, all right? And a lot of things that we're expecting from students in the lab, especially undergraduate students, is come up with very fresh and new ideas on how to make things feasible for the rat. Of course, we don't want to be crazy, but challenging and interesting. And through this, learn something new about the neurons and how they code for space. So I'll give you a couple of examples, very simple. So the first one is that we have that little robot that we manipulate. It's called Sphero. And you can see here, right? Again, I'll show you a movie in a second. And this solved a problem that everybody was having for a very long time, is can you control the speed of a rat? Can you train a rat to go slow or to go fast from point A to point B? The answer is no, right? You can try on your dog, you can try on your cat. You can train them to go from A to B, but you cannot train them to go fast or slow. And speed is very important. It turns out for these place cells, they actually are sensitive to speed. All right, so the solution actually, it's an undergrad from UBRP who had that idea and actually wrote the entire software to do it. So UBRP, to take a commercial little toy, which is a toy for cats and dogs, and then essentially use it to train to train the rats to follow this little robot. And then we control, of course, the robot. And so we can make the robot go fast or go slow. We can make the robot go here or not there, etc. So we have full control of what the rat can do. And then, so here is the thing that this is how we solve that problem. We essentially make the rat go into a figure 8. One of the leg is running fast, the other one is walking slow. And then we can see the difference in firing of these place cells in these two legs, right? Which is something that no one could have been done before. And we can have the rat do other things. We can have the rat spell the letters of our lab by walking around. So CNL, this is an actual rat doing this, or making some interesting pattern. Again, the goal is to challenge the system and to make sure that we get the data that we need in terms of trajectory, speed, etc. All right. So here is another one that we published that, of course, and this is another one that is even more interesting. This is sort of the thing that we're trying to build on. Right, the classical view of place cells is simple, as I showed you before. You have a little environment. This is like a rectangle, or the rat runs around, and then the place cell has one place field, one place where it fires. All right, so what we did is take that very place cell and put the rat in our megaspace, this enormous environment at the scale of a rat. And we discovered something really new here, is that place cells, in fact, has multiple fields. This is one cell, but it fires 3 times. All right, this is another cell that fires 4 times. This is another cell that fires 5 times, one cell. Right? This was never seen before, right? So this cell have only one place field in simple task and simple environment, but they behave totally differently if you put them in a challenging, ethologically realistic, large space, which is what rats do, right? They run in large spaces. So, so we essentially are proposing that, so far what people have learned about these place cells is great and fantastic, but it's actually not real. There is actually more to it. Cells code for space in a very different way. All right. So I just want to finish it here by showing you a few of the things that we do with the rats. So I'll let this movie play, but I'll just take that opportunity to just tell you what kind of skills or what kind of work you guys will be doing if you want to join the lab. So first of all, most of the work that we need help with is behavioral experiment, essentially training the rats, understanding what learning means. How do you actually take an animal and make the animal do what you need in a experimental subject fashion? So as an undergraduate student, you will have your own rats, 2 of them, and you will create a bond with your rats. They will become your pets, essentially. So it's very important because behaviors are complex. You need to have a relationship with the animal. That is something you need to develop, an intuition for what the animals can and cannot do and want to do, don't want to do, etc. All right, so that's one. As far as the techniques, so a lot of behavioral. So we have all kind of software to control the environment, the robot, of course, but we also have design of devices. So for example, here I'm showing you something that another undergraduate actually in biomedical engineering has designed, an implant that will actually be stuck to the brain of the rat and record from neurons while the rat is running around. And this is actually 3D printed. So we have 3D printers in the lab, so you can learn that too, how to design things in 3D printing and printing them. Now, electrophysiology is the ultimate goal. We want to record from the neurons. Now, that requires that we have a great behavior. Okay, and so the electrophysiology is not in the picture right now, but if you're willing to stay a little longer in the summer, and I'm certainly hoping you will, so these projects take time, right, working with animals. Then we have, of course, postdoc and students in the lab that are able to do the surgeries and do all of this, and then you can certainly assist and learn. But if you find a good behavior, if you help us with finding some creative ways of being complex in spatial navigation, then we can move this into electrophysiology and I'll get actual data from these neurons, so these hippocampal cells in different environments. All right, of course, we have lab meetings, so you'll be exposed to reading papers and journal clubs, right? All of this is on our website. If you want to see what kind of papers we read recently, we have a whole list there. Feel free to go look. And then you'll give a presentation, of course, and interact with the other scientists in the lab, undergraduate students mostly, and then some grad and postdoc. All right, I will stop here and answer any question you might have.
All right, students, your turn. If you have any questions, feel free to ask them now.
Hi, I don't have a specific question, but I just wanted to say that your lab sounds really interesting.
Thank you. I appreciate it.
How many rats in total do you have?
It varies. So we have between 8 to 12 rats. Typically, we work with a rat for 3 months, 3 or 4 months. They stay with us, and you interact with them, you train them, you make them do tasks. Sometimes more if they're really good rats, right? We keep them longer. Yeah. We give them names, by the way. So every rat has a name.
Oh.
Yes, it's important. Again, it's to get this sort of rapport. It's very important to get this sort of relationship between the experimenter and the rat. You have to know your rat. The rat has to know you. You have to be comfortable for them to do things like going in 3 dimensions, which is this example here. So we're trying to look at these place cells when the rat has multiple dimensions, or go into this very scary environment. Rats are very scared of large spaces, right? So you need to be able to train them to do complex things in an environment that they normally would not go into. Remember, those are lab rats, right? They live in little cages. So they have to go from little cage to, the desert, right? So again, you need to have that sort of bond that is very important.
All right, any other questions from students? All right, well then I think for now we are good. So
Thank you
students,
very
don't
much.
forget to contact anyone who's here, and thank you, Jean-Marc, for being here. I appreciate it.
No problem.
So I hope you all have good luck in finding a perfect mentor pair match for you. And stay tuned. We're having another session for conversations with faculty tomorrow as well. So thank you all. We'll see you later.
Thank you.